The scientist I chose to learn more about is the American biologist David Baltimore, who was born on March 6 of 1938. This is his early life. His interest in biology began when he was in high school and spent a summer at the Jackson Memorial Lab in Bar Harbor, Maine. That is where he first experienced research biology and really got to see the research biologist at work. After high school, he then proceeded to go to Swarthmore College in Pennsylvania and graduated in 1960 with a degree in high honors in chemistry. Baltimore originally began with a major in biology, but then switched to chemistry later on to carry out a research thesis. The following summer, he spent at the Cold Spring Harbor Laboratories working with Dr. George Streisinger, and the experience of working with and watching Dr. George Streisinger led him to molecular biology. His next stop was Massachusetts Institute of Technology in Cambridge, Massachusetts, where he took courses to work towards his Ph.D. in biophysics. He left the Massachusetts Institute of Technology to study for a summer with Dr. Philip Marcus at the Albert Einstein Medical College and to take an animal virus course at Cold Spring Harbor once again. He finished his Ph.D. in 1964 at the Rockefeller University in New York, New York. Baltimore had one brother, Robert, and spent most of his free time around his family or at biology camps with his brother as they shared many of the same interests, such as virology. He was raised observing Jewish traditions, which kept him occupied, as his father, Richard Baltimore, was an Orthodox Jew and his mother, Gertrude Lipschitz, was an atheist. He married Dr. Alice S. Huang in 1968, and they have one daughter together, Lauren Rachel Baltimore. My lifelong goal has been to help cure cancer. That's a huge goal, but even a little breakthrough can make a huge difference in the long run. This connects me to Baltimore because he is a part of the research that involves T-cell receptors that target mutations in cancer cells to help pinpoint specific cancer cells. By doing this, he is also trying to eliminate cancer, and that is something I look up to. The current field Baltimore is working on is microbiology. He is focused on the idea of viral replication and its impact on cell metabolism and hopes to continue his research in finding a way to eliminate cancerous cells, and helps with the AIDS epidemic. Published on November 10th of 2022, David Baltimore helps in writing of a scientific article titled Non-Viral Precision T-Cell Receptor Replacement for Personalized Cell Therapy. The results of the experiment demonstrated the feasibility of isolating and cloning multiple TCRs, which are a group of proteins that are found on T cells. They recognize mutational neoantigens. The T cells only react with a mutation presented by a specific HLA, which are antigen protein markers on most cells within the body, which makes it a safer target to get redirected to cancer cells. The techniques that were used to make this happen include a precision editing technique such as CRISPR-Cas9 to take out the endogenous TCR chains and insert the transgenic TCR using the TCR promoter. This data shows 3 levels of patient given different doses of T cells, with DL1 being the lowest dose, and shows the persistence and expansion of the percent levels in the transgenic T cells that were infused into them. The T cells were used to express the T cell receptor to recognize a tumor-associated antigen that selectively kills tumor cells. Here's another photo of how the T-cell receptors work within the patient. TCRs are infused into the patient and they replicate within the patient and latch onto a tumor to mark it and then attach a marker onto it to allow the cancer cells to be identified and killed. Baltimore is most known for his discovery with Howard Temin of reverse transcriptase. It is also essential for the reproduction of retroviruses such as HIV. They were awarded the Nobel Prize for this discovery. In 1986, his laboratory discovered the transcription factor NFKB, which was initially thought to be only expressed in B cells, but now is shown to be a member of widely expressed regulators in transcription. The transcription factor is involved in inflammatory and immune responses, as well as in the regulation of expression of many other genes related to cell survival, proliferation, and differentiation. Baltimore's recent studies focus on the control of inflammatory and immune responses, the roles of microRNAs in the immune system, and the use of gene therapy methods to treat HIV and cancer.